Good morning, class. Today in this lecture talk, I'll be discussing with you animal biotechnology paper growth factors. Okay, that is interleukin. Now I shall begin with interleukin. Okay, interleukins are group of cytokines which are secreted proteins and signal molecules that were first seen to be expressed by WBC, that is leukocytes. Okay, the function of the immune system depends in a large part on interleukins, and our deficiencies of a number of them have been described. All featuring autoimmune diseases are immune deficiency. The majority of interleukin are synthesized by CD4 T lymphocytes as well as through monocytes, macrophages, and endothelial cells. Okay, they promote the development and differentiation of T and B lymphocytes and hematopoietic cells. In short, this interleukin known as IL, right? So IL receptors and astrocytes in the hippocampus are also known to be involved in the development of spatial memories in mice. The name interleukin was chosen in the year 1979 to replace the various different names used by different research groups to designate interleukin 1. Okay, like lymphocyte activating factor, mitogenic protein, T cell replacing factor 3, B cell activating factor, B cell differentiation factor, and Heidikine. And interleukin 2 as TSF, okay, many more. This decision was taken during the Second International Lymphokine Workshop at Switzerland during 27th to 31st May 1979 in Ermatingen. The term interleukin derives from inter as means of communication and leukin derived from the fact that many of these proteins, that is in different types of interleukins, are produced by leukocytes and act on leukocytes. So the terminology given as interleukin. Okay, inter, that is the communication, whereas the leukin, which is derived from the terminology leukocytes, based on their act. The term was coined by Dr. Vern Paetkan at University of Victoria. Some interleukin are classified as lymphokines, lymphocyte produced cytokines that mediate immune response. Okay, now let us go through here interleukin 1, sometimes called lymphocyte activating factor. Okay, in short, LAF. It is a group of 11 cytokines which plays a central role in the regulation of immune and inflammatory responses to infections or sterile insults. Human interleukin 1 precursor molecule has 569 amino acids, the mature molecule having 552 amino acids and 17.5 kilodalton molecule weight is produced by removal of the signal polypeptides. Interleukin 1 has two forms, interleukin alpha and interleukin beta. These are most studied members, okay, because they were discovered first and because they possess strongly pro-inflammatory effect. They are encoded by different genes. These bind to the same receptor and have very similar biological properties. So they were studied most. Interleukin is intensely produced by tissue activated macrophages, monocytes, fibroblasts, and dendritic cells, neutrophils, but is also expressed by B lymphocytes, natural killer cells, and epithelial cells. These cytokines increase the expression of adhesion factor on endothelial cells to enable transmigration of immune competent cells such as phagocytes, lymphocytes, and others to sites of infection. They also affect the activity of the hypothalamus, okay, thermoregulatory center, which leads to a raise in the body temperature. Hence, interleukin 1 is called an endogenous pyrogen. Besides fever, interleukin also causes hyperplasia, in which is increased pain, sensitivity, vasodilation, and hypotension. Interleukin 1 is a dual function cytokine, means it plays a role in the nucleus by affecting transcription. Apart from its extracellular receptor mediated effects as a classical cytokine, it is also known as hematopoietin 1. In general, it is responsible for the production of inflammation as well as the promotion of fever and sepsis. Interleukin 1 alpha inhibitors are being developed to interrupt those processes and treat diseases. It possesses metabolic, physiological, hematopoietic activities and plays one of the central roles in the regulation of the immune response. Okay. Now let us go through interleukin 1 beta. It is synthesized as precursor from protein only after stimulation. In contrast to interleukin 1 alpha, its expression is induced by transcription factor NF-kB after exposure of innate immune cells to animals. This occurs, for instance, after exposure of macrophages and dendritic cells to lipopolysaccharides, in short LPS, which binds to TLR4 and acts as pathogen-associated molecular pattern, which is another group of allergens. Interleukin 1 beta are two interleukin 1 receptors. Okay, in that type 1 and type 2 receptors, the type 1 receptor is widely distributed on many cell types, and there is some evidence that interleukin 1 signaling may occur wide type 1 receptor only. Interleukin 1 has several functions, including activation of T cells. It promotes maturation and clonal expansion of B cells. Interleukin 1 is an important component of inflammatory responses. It increases the expression of cell adhesion molecules on endothelial cells. It acts as hypothalamus in the process of body temperature regulation. Interleukin 2, it was earlier referred to as T cell growth factor. It is a protein that regulates the activities of WBC, like leukocytes, often lymphocytes, and are responsible for immunity. Interleukin 2 is part of body's natural response to microbial infection and in discriminating between foreign, that is non-self, and self molecules. Interleukin 2 receptors which are expressed by lymphocytes. Human interleukin 2 precursor has 153 amino acids. The mature molecule of 133 amino acids and 15 to 20 kilodalton molecular weight is generated by the removal of signal peptide. Interleukin 2 is a globular protein with 6 alpha helical regions. Interleukin 2 signals through the interleukin 2 receptors, which is a complex consisting of 3 chains termed alpha, beta, and gamma. Okay. Further, interleukin 2, in that the gamma chain is shared by all members of this family of cytokine receptors. The interleukin 2 receptors, in short IL-2R alpha subunit, has low affinity for its ligand, but beta chain gives the more affinity to the receptor. Mutations in the gene encoding the gamma chain are responsible for the X-linked severe combined immunodeficiency, in short X-SCID, in humans. This lethal disease is characterized by the absence or very low number of T cells and several depressed cell-mediated and humoral immunities. Interleukin 2 has key roles in functions of the immune system, tolerance and immunity, okay, primarily via its direct effects on T cells. Now let us go through the therapy. Aldesleukin is a form of recombinant interleukin 2. It is manufactured using recombinant DNA technology and is marketed as a protein therapeutic and blended as pro-leukin. It has been approved by the Food and Drug Administration, that is FDA, and in several European countries for the treatment of cancers. Okay, that is malignant melanoma, renal cells cancer in large intermittent doses and has been extensively used in continuous doses. Interleukin 2 has a narrow therapeutic window and the level of dosing usually determines the severity of side effects. Okay, now let us go through the clinical research. Interleukin 2 has been in clinical trials for the treatment of chronic viral infections and has a booster, that is like an adjuvant for vaccines. The use of large doses of interleukin 2 given every 6 to 8 weeks in HIV therapy was found to be ineffective in preventing progression to an AIDS diagnosis is to large clinical trials population trials which are published in 2009. Okay, so this is all about the interleukins. Hope you have understood. Kindly go through, make a note and get ready for this particular interleukins for 5 or 10 marks. Okay, make your own notes and prepare for this interleukin. I, Dr. Pratibha KS, signing out for today. Stay tuned with me for more videos. And thank you for watching.